Thank you so much for the kind introduction. Yeah, I'll go to talk about PentaBase and 4 topics that I was given. And the 4 topics, which is how did we do during COVID-19? It was also wanted to hear about public, private or private-public relationships or innovations and partnerships. Research in private versus research in the public. I don't have that much experience in research in public though, but I'll try to do something. And then I was asked to say how to create big business. So before I jump into that, I will tell you a little bit about myself. I founded my first company back in 2001. This company was founded together with 2 friends that I got to know at the university from 2 other departments. So one from marketing, or he was Kent Merck, and one from molecular biology. Because I always believed in that doing chemistry, we could do something real to change lives of humans. And in order to do that, I needed to get closer to applying the chemistry closer to the patients. So this was what we tried to do during my studies here, and under the supervision of Erik Bjergård Petersen, who is in the audience here. I invented a DNA technology called RNA. I'll come back to that a little bit later. But based on that, I founded my first company. Had that for 2.5 years before we sold it to an Australian company. I went there with my key employees and worked there for 1.5 years, presented them for a new idea I had. At that time, they didn't think the idea was applicable now. I mean, they thought it was a good idea, but they thought it would take many years until it will become real, that it will be used by hospitals. So I returned and founded PentaBase in 2006. Just to say that I have done a little bit of research. I have 12 publications and I've written 9 patents. But in many years, when you are a business that do not have much funding, you don't focus on writing either publications nor patents, except the most important ones. PentaBase is a science-driven company, so we really like science, and we would only do things that we think is scientific valid and has a high impact, that we create things that are better than what is already on the market. It's founded in 2006. I started in my utility room in Skalbjerg, where I lived together with my wife. And we shared the same plug because there was not much electricity in the house. So every time I had to do HPLC, she couldn't do washing of the clothes or anything. Not that it's always her that did that, but we had to share that. So quite nice. Then I moved to some toilet facilities in Søndersø at the Erhvervspark. There was a collection of companies there. They had an extra toilet building where I could remove the toilets and install a laboratory. So I moved to that, and then I moved into the harbor in Odense, Odense Erhvervspark, which is HK that is driving that, and they also help by installing my laboratories and that, and then in 2021 or 20, we at the end of 20 we bought a new facility in Odense C, just behind Biltema and Plantorama, where we now have approximately 3,000 square meters. I have in total 110 employees. And 7 laboratories in Denmark. And the 6 others, the ones that are not in Odense, are mostly corona test facilities with own labs. But they also do some science and try to develop new things that we can offer for the public in general. My mission is that we would like to use our DNA-based technologies to improve diagnostics in general, especially genetic diagnostics. We believe that every patient is unique and that they need to be treated individually. So basically, I would like to get rid of what's called cancer packages. Today, if you're diagnosed with cancer, you are met with a package. So already from the beginning, they know that you should have 8 treatments of chemotherapy, for example. And then they look at it again. Of course, every 60 days, they do a scan to see how well it goes. But they only stop the treatment if you have severe side effects or there are clear signs that the treatment doesn't work at all. They cannot start a new treatment if they think, oh, now there's a new and better drug. Let's stop this here and try the other one. What we would like to change is that we constantly improve the treatment of patients, and it's not easy. Drugs are approved through a clinical trial where they say you should follow this protocol, this many treatments, this amount of drug, and then it's approved on these bases. What I'm suggesting is that we should monitor patients and see, oh, maybe you should have 20% more of this drug, or maybe it's actually working so good that we can reduce the amount, or maybe it's not working at all, so we should change. So this is a big ambition. One thing that we say is more important than having a mission and a vision is actually having a good culture. And so that's why I've stated our 4 values. So these 4 core values is something that my employees and I agree on. This is very, very important for us. And it's not one-way values. I mean, so when we say accountability, that means that if I promise my employees something, they can trust on it. If I promise my customers something, they can trust that we do our utmost to deliver that. But also the other way around, of course. I expect that I can trust my employees that if they have said they want to do this, they'll do it, right? Adaptiveness or adaptability is that I cannot foresee the world. I couldn't foresee Corona before it was nearly here in Denmark. But we need to be able to change and meet these new things that comes to us. And this is the adaptability. But it's also that we need to, if employee gets pregnant, of course, that we change the way that he or she should work. In this case here, not in the lab, for example. We find a new job, a new thing that could be done, which is also interesting for the employee. Caring is caring about each other. It's also caring for the patient. So whatever we do, we have the patient in mind at the end. What we do should be better for the patient. I'll come back to why I state this so many times. And then scientific excellence. We are a bunch of nerds. We really like our science. And the thing that makes us really, really happy is if others like the products we develop. So we often are so scientifically correct that we tell also all the downsides of our products or what are the limitations in that. And therefore, we are really bad salespeople. Let me start by the travel through COVID-19. So when it started, and for us, it started already in January. That company that I sold my first business to, a year after I sold the business, a year after I left them, after I presented my new idea, they called me again. Ulf, by the way, the idea you presented for us, could we try it? They tried it and they became my biggest customer again. So that's one learning that I could say, that never burn your bridges. I left there on good standing. I actually bought back all the equipment that I sold them in the first instance. And then they became my my biggest customer again at that time. But they started ordering a lot in January. Actually, I had to work nearly during the clock around the whole day from January already. And I was nearly not at home because I had all in all 12 employees and 2 students. And that was covering our oligo production, our administration, our assay development and design, our product quality and quality control and what's called quality assurance. So we were basically 2 people in each department. One of them was my mother, of course not paid, and so we really used all we could. I mean, to have this company just running with 12 employees and 2 students. The year 2019, our revenue, that means we sold products for around 1 million euro at that year. And I still had 12 employees, so it means that there was 500,000 kroner per employee that year, and we actually earned money. So we're running on very low burn. I say the average standard in the biotech community is, you say that 1 employee costs 200,000 euros per year. So this was our setup. So now orders just came in. And of course, if you're really used to being hungry, you'll grab every order there is. So I was working, I took the night shift, I took the beginning of the day shift, then I slept 2 to 4 hours in the basement of the company, and then I started again in the afternoon producing. So this was basically how it was. At some point in this, I think it was in February, I stopped up and I took a weekend off, said now I need to go home, see the family and do some reading. And there I spent the Saturday reading about this coronavirus and that. And I tried to find Chinese literature written in English about what it was. And then I realized this would come to Denmark. So we knew that this was going to come to Denmark. We knew that it was big business because our biggest customer, which was placed just next to China, ordered a lot. So we were sort of warned. At this point here, I ask people to be very flexible, to work as much as they can, because actually my first thought was we're going to be closed down. Mette is going, our prime minister is going to close the country, so we are not being allowed to go to work, which means that I can't produce for Australia, which means that I'll have to sack half of you if this lasts for half a year, which was what people thought at that time, right? So this was sort of the first thought. Then I thought that, okay, maybe we would be allowed to work, but a lot of other companies would have the same destiny. So they would have to go bankrupt, or they would go bankrupt if the society closed down. And knowing how difficult it is to start a business and how much you fight for it, I actually thought, now I must help. I must do what I can to see if I could help. So this was when we realized we had a duty. We worked round the clock. I tried to say to point out that we could help Denmark as such. One night when I was working in the lab producing for Australia, the news were on, and in that news there was 2 stories. The first story was about Corona and how countries shut down around themselves. and focused on themselves. And the next story was about that Denmark had everything under control because our health minister had ordered 5 instruments from Switzerland, so they would help us. And basically, I got very upset. First of all, thinking that Switzerland would come and help Denmark, at the same time. And then, second of all, what I heard was that the health minister said, we have ordered 5 instruments from the biggest competitor of PentaBase, installing instruments in all hospitals, which means that I would not have anything to sell when Corona is over. Because it was Roche, and these instruments were fully automated. And after Corona, they have to justify why did they spend these millions of euros on buying these fully automated instruments. And they would, of course, need to run cancer diagnostics on them afterwards because there was kits for that. So not only did I see that they didn't want to listen to us during the Corona, but I also saw that they wanted to remove my future income in this. So that was why I got a little bit upset. We had some advantages, though. There one main owner in the company, which is me, and I am willing to take risks. We also have employees that were very flexible, adaptive. And so one of the first things I did in these meetings I had the 2 daily meetings. I had one in the morning to tell the people that were coming in the morning what had happened during the evening and night, and then I had one in the afternoon to tell the people that met at that time to say what had happened through the day, because so many things happened. But I empowered them also, so all employees were allowed to buy whatever they needed to do their job. So no improvement was needed. And this was very important. This was actually, I think, the most clever thing I did at that time. Because when people realized, when the employees realized, and I don't forget that day, I was having these 2 speeches here, and I think it was 10, 14 days after Denmark shut down. That was the first time when they realized that they were the experts. The Danish government has for, I don't know, 4th time said something crazy. And suddenly they realized what the State Serum Institute was doing in their Corona test. I'll come back to it a little bit later, but basically what happened in Denmark was that we threw away all the scientific knowledge that we have built up through years. We ran the Corona test in the public system sort of like a hazard. One target only. When you have virus that mutates this much, you always have at least 2 targets. We didn't have any internal control, so if people didn't, if the swapping wasn't good enough, there was no way to tell. I, as a private company, could never deliver the quality that was delivered from the public sector here. So when my people realized that, then things suddenly changed. And it's not easy. We are raised with having trust in the public system. We all think that what happens is for the best. There's a reason for if they take this decision. So, we realized that we were experts. We needed to do something. So in March, we launched our first assay. This here is basically we couldn't deliver to the public Denmark. We didn't either collaborate with them, but at some point Mette called Novo Nordisk to ask if Novo can help with doing testing of Corona patients, which is obvious. I mean, they produce insulin, so why not? Novo called us and asked if we could help. And we developed an assay with them. They came up with a few things, that it should be exactly the same as CDC in US had asked for. It should be multiplexed, so their CDC's assays were a simplex reaction. It should run on instruments that was no longer used at hospitals, because we couldn't compete with the plasticware for hospital things, so it should run on really old crap. And then we should use their supplier for master mix, because Novo believed that they were the only company who could actually get supplies in through these days. It wasn't easy. We managed, and when I say we, it's mainly my Chief Scientific Officer, Rasmus Petersen, who's also educated here. He managed to make one, convince them this was exactly as good. We had 100% correlation with the CDC. Then we made our own assay. It took only 2 weeks to make that. It was a lot better, a lot faster, easier, and it took another 2 weeks until Novo was convinced that this was actually better to use than all the things that they have set up. Also using our own suppliers. In December, we improved the speed of the assay, developed something called COVID Detect Fast. This was actually the reason that we, as the first one, could open an airport in Europe. So Frankfurt Airport was the first airport that reopened in Europe, and they used our assays there. And then suddenly things went fast. We also developed others. I stopped here in June 2021. We have since developed what we call COVID Detect Ultra, where we detect coronavirus using PCR in 23 minutes. And we can detect hundreds of those. So it's on ready available instruments in laboratories. We also look at the results, and I have 2 of our largest customers here. So they were doing PCR tests twice a week. And when this is very interesting, it's because it was exactly the same as schools did, but they used antigen tests twice a week. What we saw in these companies was that the average infection was 87% less in the companies as it is in the society around. So by making sure that your colleagues were not infected, you didn't get infected at work, and that reduced the risk of you getting infected with 87%. So basically, no shutdowns, no internal infection chains. The 2 companies here both used it for building, expanding their facilities. PM Group is building for Facebook, the data center out here. So no delays on these constructions. And that was in contrast to what you hear with all the public hospitals that are being, the super hospitals that are being built. They've all been delayed a year or 2 due to Corona, and cost 1 billion kroner more or so in average. So what did we learn from that? Yeah, we learned that governments, they could set their own standards, which was below, substandard performance rules. Also that these rules changed after political influence. Suddenly, first antigen tests didn't work, then suddenly they worked, then were the solution for everything. Then they kept using the antigen despite that they said we won't go back. Then again, there was a surge in infection, and they went back to antigen tests. And there was no transparency in the rules. I could say that, for example, we have developed a saliva-based corona test, and we say, what's the big thing about that? The big thing is that you can take the sample yourself. It works as good as our throat-based test, which is approximately 100 times more sensitive than the one used in the public. Now you could move the sample and not the patient. So you could move the elderly. They don't need it. You don't need to send out a car with a nurse swapping those. They could make the samples themselves. People on boarding schools, they don't have a car. They can't drive to a test center. They could send the samples if they're afraid that they were being infected. People that works on shifts, now you could just collect over a full day, just transport one sample at a time. You don't have to come multiple times to the same company. So many things could have been improved just by that. But instead of embracing such an invention, they actually made a rule that you could not use the data from a saliva-based PCR test. You couldn't trust it. And we have peer-reviewed the article now and published. So this was basically saying we want control. So there's no scientific data. When I had the discussions with the health ministry, the only thing they could come up with that the reason for using antigen tests were availability. But they spent more than $1 billion, euros, on antigen tests. Of course, there were many that would like to supply it. Then we heard a lot that Denmark did very well through the corona. Did we? Anyone? On what parameter did we do well? Financials? We threw a lot of money out into the society, yes. Nobody went bankrupt at that time, but what about our inflation now? Did it do that we didn't get corona? Nah. Actually, Danes are the people that have the highest infection rate of all in the world. And this is not because, it is because we've tested a lot. It was because 3 out of 4 that actually carried corona was told that they didn't when they tested in the public system with the antigen test. That was why we spread this virus like no other country. But our hospitals didn't close down, and this was then the parameter that they held as a success. I don't know in which country in the world the hospital system shut down. I haven't heard about it. But this is the parameter that sort of is highlighted. But if you know the hospitals right now, they're really stressed. There's not enough employees. They have not had vacations. They have done overtime. There's still a lot of corona patients filling up the hospitals, and we are actually just coming out of summer. So I don't know on which parameter that it is, but what if we only had 15% of all the infections that we had? How would it then have looked? I don't know. The next topic that I would talk about is the private-public partnership. And remember, I come out of a history where I really don't believe in all that's just public. But I really believe in that we could do a lot more together. But it's difficult. And why is it so difficult? One of the things that is difficult is, me personally, I believe that I'm better in doing everything nearly than anybody else. But then I experience that somebody actually knows more about stuff than I do. And when I've let go and let people work together, I see that the results are better than what we could do ourselves. I also see that we have not been good at implementing automation. We haven't been good in digitalization because we insisted on doing it ourselves. We could have done better on those. But I also believe that universities has the same tendency. When we talk public-private innovations or public-private partnerships, universities as such think, what can I help the private sector with? This is sort of the standard thing. It's seldomly that the university thinks, maybe I could get a private to help me with this. So this is really the things that I would like to change. One of the things that I think we should do is, first of all, to define some projects where we have a common purpose. For example, if we look at personalized treatment, which is what we aim for in our vision, then a lot of different players could have a lot to say. The general practitioners, they have the contact with the patients, right? They would be very important in being able to tell the patient what have we found, why are we changing this and that. So very important in such a thing. University, you come with all your innovations. You could see things from various levels. You have so many different techniques. You're not a PCR company like PentaBase. You have everything, Raman spectroscopy, mass spec, so many variations of techniques that you could think of and bring to the table. Hospitals, they know what current standards are. How do you do with current protocols and that? The society, they could put up sort of the frameworks, the regulatory changes that needs to happen if that should ever have success. And then the private sector, we could bring in the ability to scale production, to reduce costs, to look at the workflows, such. But there are some challenges to overcome. And one of the challenges is that we look at each other and think we understand what the others will. It's a common thing that companies want to earn money. So this is the only reason why they go into such a project. And yes, I really like to earn money, but not for every price. So during Corona, we never sold a single antigen test, despite that we were by far the biggest company in Corona because we started out as the first. But we didn't want to sell this technology because we didn't believe in it. It wasn't scientific excellence. We didn't sell. I had 5 brand new instruments on stock when Magnus Heunicke announced that he had ordered 5 from Switzerland. I had an order coming from a German company on our website for 5.5 million kroner, so 0.8 million euros, nearly the same revenue that we had had the whole year before. We called them and said, sorry, we know that we have it on our webpage, but we cannot deliver because we don't know what Denmark would like us to do. It wasn't about money. When we developed for Novo, we took 100% of the risk, didn't ask for any money, didn't ask for anything. When they approved the assay, we sat down with Novo and we did a calculation. Novo said at that time, we believe we should test up to 6,000 people a day and Corona would last for 3 to 5 months. We did a calculation and I said, I just need to be able to have my employees by the end of this. So we ended up at a price that said 25 kroner per reaction. It turned out that we tested a lot more than 6,000 a day. It turned out that Corona lasted for a lot more than 3 to 5 months. So we earned money also on Novo Nordisk because when they had to transfer, they called us and asked if we want to take over. So Novo Nordisk was doing all the tests that the hospitals couldn't do. So when the hospital didn't have capacity, they sent all the extra samples to Novo Nordisk, and they did the test there. They asked if we wanted to take over. We said yes. 24 hours later, they found out that we were a private company. So we couldn't do that. So it was transferred to DTU. So we helped DTU set up this and they got the same agreement. But in the transfer, Novo Nordisk ordered 782,000 reactions. And suddenly I earned money. Wow. I also, a month after I found out, after we asked that, I called that German company again and said, Denmark don't want our machines. Would you still like them? Yes, yes. So we exported, and we exported 75% of all our revenues came from exports. And this was the reason we helped open airports like Frankfurt and that. Okay, so it's not just about earning money, but we need to build trust to each other. And sometimes it seems easier to build trust with people that are far away. So with all our cancer developments, we found a hospital in Switzerland with whom we collaborated very well with. There was no questions. I mean, this was just a scientific collaboration. We didn't think of each other as competitors or competing for the same employees or money on that, which I feel is an obstacle for doing private-public collaborations. And sometimes also it's difficult because who get recognized for a successful private-public collaboration? Often it's the private companies because that's a better story in the news or whatever. So there are some things that we need, some challenges that we need to meet. I believe that the solution will be partying, getting to know each other, having talks where we can find out what is it that we actually really want, and then being open about the aim of the private-public partnership and who gets what out of it. Also talk about the financials, publications, patents, and that. And that said, I also say a lot of lawyers and that because then we make agreements and that. But we need to find a way where we can sit down and agree on this actually makes sense for everybody, and then have the paperwork to come easy afterwards. That was just a few words I had on private-public innovations. There's also private versus public research. So when I look at it, I don't envy public research because today you nearly need to find your own money to be allowed to go to work. So you need to apply for so many grants and that. I don't know why that is. How can you ever be free to do your work, focus on what you're best at, if you should also do applications the full time, describe how the market is at the things you develop at the end and that. I guess that the Rio here is larger than any of the groups that are research groups that are at the university because you need so much help. I really don't envy that. But what I do envy is that you have access to each other, different departments. You can easily make integration of projects. You can make things where you take the best brains of this and that. That's very difficult for a company like us. I might be able to attract some really good scientists, but it's more difficult to attract a really good bookkeeper or a really good doctor, because that's not what we are known for. People can, scientists, especially within genetic science, can maybe easily see themselves in PentaBase, but it's more difficult for maybe a marketing expert because they're used to seeing campaigns in Denmark. How does this coffee brand position itself against other coffee brands and that? And now you have something very different. So this is the difference. One thing that we do in the private is also we constantly see if the project is going the right way, so we can stop a project when it doesn't make sense. But you get the money for a predefined project. that you applied for 2 years earlier or that. And if you want to keep your employees, you need to finish that project, which is, yeah. PentaBase was based on RNA intercalating nucleic acid. So this was a fantastic invention that Bjergård and I did back at the times, and actually, yeah, I don't have time to go into this, but this was the basis of PentaBase. So I had something to start with, some uniqueness in the company. We haven't stopped doing great science. I would just highlight 2 things. One product that we call microsite, which is for looking at microsatellite instability. Microsatellites are small pieces of DNA in our genome where we have repeated one base many times, for example, 28 times. So DNA consists of 4 different bases. So if you have one repeated 28 times, it is very difficult for the enzymes that copy when a cell divides, that copy to make sure that they have exactly 28. Therefore, we have a repair system that after the DNA is doubled, so now we are ready to make a new cell to divide, we have DNA repair systems that check if it's a true copy of the first. If they don't work 100% efficient, mistakes will happen. And mistakes will happen very early in microsatellites because it's very difficult places to amplify. So it's an indirect measure for if your DNA repair system works well. Microsatellite instability is also the biomarker for if you can have checkpoint inhibitors as a cancer patient. And checkpoint inhibitors are drugs that helps you boost your immune system to combat your disease. And there are examples where people have said you have a few weeks to live, but we try this drug and they end up being cured. So for the people on which it works, it is a wonder drug. And it is that because you get your own body to combat the cancer disease. We have reduced the time it takes to get an MSI status from 2 days to 2 hours. And we have also removed the subjectivity that where you need as a doctor to sit and evaluate if 2 hand structures are alike or if they are different. I don't want to go into the technical thing. There's a little on the paper up here. But this just to say, and this is very useful. This can mean that a patient start out with the right treatment instead of starting out on the standard chemotherapy and have to go through all 8 cycles before they then can try the drug that might have cured them from the very beginning. So this is really a thing. The other thing I'm going to mention is our newest invention called EpiDirect, which we are very proud of. EpiDirect is the first in the world where we can directly see on a sample how much methylation has happened on the DNA. And for those who don't know with methylation that, when we are born, we're born from an egg and a cell. These cells divide. How come it turns into eye cells, liver cells, skin cells, and that? What is it that makes the difference? The difference is that some genes are expressed in certain cells and not in others. And this is happening via epigenetic changes. So and then the other thing is when I was 18, I had a health that was a lot better than now when I'm 48. Why is that? I have the same DNA. But it also happens over time that some genes are shut down due to these epigenetic changes. So, and methylation is one of them. Today, what we do is we take out a sample and then we treat it with some chemicals, so we change these epigenetic changes into sequence differences. The problem with that is the answer becomes either 1 or 0. Now, either you have epigenetic changes or you don't. Another challenge is that at least 80% of the sample is degraded during this chemical treatment. And it again takes a long time. So what we've been able to do is to quantify the amount of epigenetic changes that had happened in a real-time PCR experiment. In less than 2 hours, we can say if a patient is having these epigenetic changes and how much. And I don't even know where this will lead, because now we might have the tool to look at the way till we get age-related diseases. So maybe we can find out how do we get Alzheimer's. Are there things happening that leads to Alzheimer's at the end? And maybe at some point we could be able to reverse it, so we never get the diagnosed Alzheimer's, but we just had a step on the way which we reversed. I don't know, but at least the tool that we have developed here, I am extremely proud of it. And we have very, very high hopes for that this can be one contribution to changing diagnostics in the future. So the last thing I was asked to talk about is to creating big business. And that, I mean, the challenge to go from being a scientist to having a big business, it's huge. And actually, I would say the most important thing is the bottom, which I said you have to be naive. I mean, if you knew how difficult it would be, you would never have started. But that's one thing. If you have somebody you can trust that is on the way, it gets easier. And you have to be agile. I mean, it's not easy to earn money in life science. So if there is someone who is willing to pay for something, do that. In Denmark, when we are in life science, we often have one customer. It's the public system. So if you don't get the public system as a customer, you don't have a customer at your home base. So this is a challenge. 95% of our sales before 2020 was export. So you need to be able to network and get international business going as well. So this was my last slide. So finally, I would like to thank you for your attention.
So thank you very much, Ulf, for the very nice overview of your career and over your success about your business. And of course, and we also heard about exciting the science that you have built up in your company. So is there any questions? And actually, I have one question. And you said that and you have 50% of your employer actually from SDU, right?
Yes.
And you also have some interactions with the university?
Yes.
And do you have specific examples, and how do you cooperate with the public institute like SDU?
I mean, we first of all, we'd like to have much more interactions with the SDU. Already in 2019, I had 17 students making projects in PentaBase. And the reason for that was that then I could select the best talents among that. I selected 2 that were employed right away. One of them, she is the mastermind or the green fingers behind that EpiDirect there is. So the access to talent is really, really important. But we'd like to, I mean, collaborate, make science, get input from the public world as well. What is it that they actually need? The other way around also, what can they offer? What can we purchase from them? What can we do in collaboration? We love to do a lot of that.
That's great. Any questions?
I just wonder, you know, the last project you're talking about, have you tried to engage with some doctors or, you know, in terms of trying to get this out and functioning?
Yes. We have one CE-IVD marked product based on that technology for a gene called MGMT. Because if this is the only example we could find where this methylation actually led to an interaction or with patients. So for MGMT in glioma patients, they could get a special drug if they have these epigenetic changes and their prognosis becomes a lot better. But we are looking for hospitals and that to do a lot more collaboration, research and development with. And we have now succeeded with a few Danish hospitals as well. So it helps that we have become a larger company to do local collaborations. Definitely.
Yeah. Just an add-on question. So if I understood you correctly, you were saying that you sort of were sitting on a technology during COVID that wasn't really used in Denmark. And do you think there might have been a way you could have lobbied stronger or would have changed anything you did in relation to the authorities to make them understand that?
Yes, you use a very, very correct word, lobbying. What I found out is that it is not much about the science you offer and that it is who you know. I was naive. I thought that the best solutions for Danes would be what would be selected. I thought that if you came with things that were cheaper and better than what was used, they would of course listen to you, but they didn't. I would say that the 25 kroners we sold per test, that is around 25% of what the public was paying for the reagents to do a PCR test that didn't have internal controls, that only had one target and not three, where the answers came within 48 to 72 hours instead of 4. So basically there was no reason, scientific reason for not to do it. But the public has an eye on protecting public institutions. So if you let a private company do what Statens Serum Institute was supposed to do, then what would happen with Statens Serum Institute in the long run? Why wouldn't I, as a company, then make influenza test or make chlamydia test or make other things? So maybe it would disrupt the way that the public structure was at the time. So I guess that's the reason.
Thank you.
So first of all, Ulf, congratulations with the big success. It's really impressive. It's also very impressive with the last product you told about. It's a long-standing goal to be able to detect these methylated bases. And I would love to be able to ask you the chemistry background on how you do it. I'm a little bit afraid that you might not be willing to tell us, but if you are, that would be great. But can you at least say a little bit about the basis for how you establish this necessary discrimination?
Yeah, and I said I believe in open science and that. So PentaBase is based on intercalated nucleic acid, and you know it. So it's some intercalators that integrates into DNA, and what it does is it increases the stacking effect tremendously of the oligo. So the electron sharing of the bases in DNA and these RNA modifications there are high. When you add a methyl group to the cytosine, this is an electron donating group. So it becomes now a higher interactions with methylated cytosine and the intercalator. And this higher interactions is actually large enough to that we can get a difference in affinity. So now we can make primers that only bind to the methylated DNA and not the unmethylated. And of course there are some tricks here, and sorry that I be very technical now, but when you do PCR, you lose this signature after the first PCR because the bases that you put into your newly synthesized strands are not methylated. So it needs to be really good, but that it can be done. I think there is a question up there.
Thank you for the great success story you told us. But this was your second company, right?
Yeah.
So what I think is usually the hardest part is making your first company, and after you succeed on that, it's much less hard to make your second one. So did you always have this mindset that you want to make a company at some point? And where did you actually get the idea for the first company? Was it something that you saw that was needed, or you were researching yourself?
Thank you for that question. Yes, I always had the mindset that I wanted to start a company, basically because I didn't think that anyone wanted to hire me. Then I was lucky that during my master's here, we made that invention that seemed interesting. And it wasn't from the scratch. We're standing on the shoulders of many here. The Institute has built up a lot of knowledge about DNA technologies. Jesper, who asked the question before, had made an even much more well-known DNA analog called LNA there. So we're standing on the shoulders of others that had helped us see that we could use this for something. My invention was the first that could differentiate between DNA and RNA. And therefore, I had something. First of all, I thought I was trying to inhibit HIV, which is an RNA, and my compound didn't work. The modifications didn't work at all. Actually, it bound very well to DNA, but not to RNA. So I thought my whole project was wasted, right? It took around 14 days to think about it until we say, hey, right, we got something here. This is very special. Maybe I could rewrite the project and then make it into some positive thing. I was then employed by Bjerregaard and Jesper actually for 1.5 years to do more research on this. And in the meantime, I negotiated with the university about who owned the things and that. I ended up taking a loan in my house to buy my own invention and start out. This was how motivated I was to start a company. Then it's difficult because then you're told that you should then get investors. I did that. I also got companies from Australia that purchased my technology and that. I ended up having to use lawyers for the local investors not to make my company go bankrupt instead of selling it to Australia. So it's not easy. You really have to be determined and that. But you can. If a lot of hard work, and then I don't know if the second one is easier. I didn't have much money. I spent it all in Australia. So, but of course I had some experience.
And it's nice that you're patient too, right? Waiting for opportunities. You said one of the key drivers for your whole company and concept is that you want to do something for cancer patients. You didn't talk so much about the approach for that. Are you targeting individual genes or panels of genes? Or how is it you want to change the way we diagnose and
what we're
treat
able to
cancer?
do, and thank you for that question. What we're able to do is from a blood sample see the amount of cancer DNA that is circulating in the blood compared to normal DNA. And because we have this special technology, I'm able to see one molecule with a single base pair exchange in a background of 64,000 natural DNA molecules, or healthy DNA molecules. So this means that the day after a person has got the medicine, we can see if it's working. And then by simple blood samples, we can monitor if the treatment keeps working or not, or if it should be adjusted a little bit. During my career, I had an investor that unfortunately got colorectal cancer, and we monitored him. It was a non-intervening study, meaning that they didn't change the treatment due to our data. But at least 3 times during that treatment, we and oncologists would have done differently had they used the data. He ended up dying after 2 years, unfortunately, but this has been a big driver also for me to see that it must go out there and be used, the technology.
Okay, so I don't see further questions. It's very nice to hear your success to bring science to the real marketer for the applications here. So let's thank Ulf again for the nice talk. Thank you.
And thank you.